So I'm going to talk today about patient-centric translational research, and I'm going to talk a little bit about what Baron was touching on in yesterday's sessions, but a lot of the ideas, or I'll talk about a lot of the ideas around image analysis and translational work that a lot of us have talked or heard about today, and specifically on how translational research and development helps us understand more about our primary systems and drives forward the technology for in vitro diagnostics and eventually leading to functional precision medicine. So it is an acute leukemia session, but since we haven't gotten together as a group in a while due to the pandemic, I wanted to share more of a general update. So please bear with me as I venture a little bit elsewhere. So my disclosures, I'm a shareholder and employee of Allcyte in Vienna. So we believe that there is a patient-centric paradigm for preclinical research and development that will lead us to a better understanding of functional precision medicine. Cancer models are widely accessible and have been the discovery engine of therapies we currently have, cell lines, animals, and organoids, but we end up with a 95% attrition rate in the clinic of drugs that ended up there. Whereas I think we all believe that a more translatable model, primary tissue in this case, is really as close as we can get to patient material, to a more relevant model system, and thus increasing the chance of trial success by putting patient at the center of the preclinical research and development idea. So just to mention that Allcyte was really built on the backbone of Exalt 1. I'm not going to go into details on this. Baron talked about it, so Taya and Philip's going to talk about it next. But just to say that having a technology and a previous version of a technology that we know at some stage is translatable for late-stage hematological cancer patients and therapy prediction is really a great starting off point for translational research. So as introduced slightly yesterday, our methodology of choice is high-content confocal imaging of primary patient material and then single-cell quantification, and we term this pharmacoscopy, which is kind of the catch-all term for us imaging patient material. And I'm going to talk about 2D and 3D image analysis here and kind of what we do with it. So the samples we take are dependent on the indication, so lymph node, peripheral blood, and bone marrow for hematological indications. Malignant pleural effusion in the ascites, so secondary malignancies with cancer cells and recruited immune cells for solid tumors, and then also now solid tumor biopsies. And I'll get into homogenization in a moment. But for the liquids, at least, we do phycal density gradients or low-speed spins, but no outgrowth or selection or organoid creation. And then we take all of our cells and we add them into 384 well, or now 1536 well screening plates, and for some indications, 1536 wells we're moving into plates. And each of these wells contains a drug of interest, a small molecule, a biologic, or a cell therapy. And then at the end of the assay, which is usually about a couple of days, we fix these monolayers in place, with these layers in place, and we stain them with antibodies or dyes. If we want to look at population changes, we look at cell surface dyes against cancer markers or other immune cell markers. If we want to look at things like viability dyes or mitochondrial dyes, so we can do that too, and nuclear dyes, etc. And then we image these plates and we detect and quantify single cell phenotypes with deep learning supported image analysis. A number of us mentioned this idea. I'll show 2 slides on it. And then we integrate single cell phenotypic data at the population level to kind of see what we're doing, either as its dose response curves or activation states. And Allcyte hopped on the deep learning and scalable cloud computing train at the very beginning. And this has really enabled us to think very big when it comes to both image analysis and also capacity. So for single cell segmentation and classification and phenotype quantification, we turn to custom deep learning driven image analysis tool chains that we've created in-house. And I want to show you an example of how we tackle some complex images. So this is actually a solid tumor sample. And in our solid tumor programs, we need to homogenize these tissues. Same goes for the lymph nodes in the hematology. But by introducing or by creating a single cell suspension, we're likely introducing too much background cell death. And so and maybe something that's more amenable to flow cytometry, so single cell suspension. But what we want to do is we want to only lightly homogenize these. And so we're keeping intact, we do get single cells. What we end up with is a mix of single cells and what we call microaggregates. These are 50 to 100 cell pieces or aggregates. I believe the cell contacts are representative of what was available, what was there in the solid tumor. And then clearly to define what cells are in here and segment down into them, we need to do 3D confocal microscopy. You can see this here. And then clearly for image analysis, we can, let's say, segment and classify these cells with really high accuracy using deep learning-based segmentation and other approaches. We believe that classical image analysis approaches would not necessarily work in these cases because the membranes are so fused together both in the 2D and in the 3D space. So as I said in the very beginning, we use translational research to understand the most we can about, one, the model system, but two, about our own technology and our pushes for in vitro diagnostic growth eventually, or into the IVD space. And so we spend a lot of time collecting technical performance for many of our indications. And so I wanted to give a couple examples of this: linearity, trueness, biological reproducibility, so things that are akin to lab developer IVD tests, also things like limit of blank or limit of detection. And it's not just, okay, well, we have these pieces of information, but it's also then, the ability for us to really leverage our data a little bit better if we understand day-to-day performance attributes or user-to-user or patient or sample-to-sample attributes. And it's not just technically understanding our platform, it's also the pre-analytic studies on the material itself. And actually, JP just mentioned this in one of the answers to a question. But many of us are on hospital campuses. Many of us have the ability to collect fresh samples from the clinic. We have a number of fresh programs running as well, but we have a growing biobank. And so do our frozen bone marrow samples, our frozen samples in general, respond the same way ex vivo to drugs as our fresh samples do? And so this is a study in collaboration with Professor Heinz Sill from the Medi Graz, where we prospectively collected 10 AML bone marrow samples. We screened exactly like we did in EXALT-1, and then froze down a matching sample and then rescreened it. And this is just cell fractions, and we can see the good linearity. It was an N of 10. And the way that we ranked drugs as well in the EXALT trial, we also see good rankings between fresh and frozen here. And I think, it's also critical to understand shipment studies for those of us who are shipping samples. Maybe this is true for AML, maybe it's not true for a different indication. And I think I'd like to see more robust pre-analytics. I think, it's challenging to set up. But I think the data does drive our field forward quite a bit, as does understanding the reproducibility of dose response curves. So this is 3 samples over 5 days and a couple of different drugs. And these are each one is a different freeze down, same user, same scope, same platform. But we can see that there's reproducibility in our drug responses as well. And so just a couple of examples of what we're using this platform for currently in hemato-oncology. So this is an ongoing program we have where we're doing biomarker discovery or genetic biomarker discovery in diffuse large B-cell lymphoma, where we're using drugs as tools to ex vivo to stratify these patients into responding and non-responding cohorts. This is an ongoing project. We're collecting 100 diffuse large B-cell lymphoma samples, which is challenging to obtain 100 of these, but we're working towards it hopefully this year. Library of drugs, patients. The darker the blue one of these boxes is the more targeted and active a therapy is against the B-cell cancer population. The more red, the more generally cytotoxic, and the white is not reactive. What we can do is we can take these drugs and we can say, okay, I have a responding cohort of samples here. I have a non-responding cohort of samples here. And then we can collect orthogonal data. And in this case, it's whole genome sequencing data and RNA sequencing data of the B-cell population. And we can say, okay, do I see patterns of the gene variants, expressed gene variants, ranked, ranking the patients by the drug response score as in the EXALT trial, and saying, here's my responding, here's my non-responding group, and do we see these gene variants? Because the genetic biomarker or the genetic point of care is still quite popular, obviously, in the clinic. And then one other example I want to show you in hemato-oncology, is our CAR-T program. And this is some first data that's just come off our platform last week that we're really excited about in our first CAR-T experiment. And this is data in collaboration with Uli Jaeger from Medical University of Vienna, where we have biobanked CAR-T samples from patients who unfortunately had died before they were managed to give them the treatment. And we have frozen lymph nodes or blood from these patients also. And we want to see if these samples or if these CARs are active in our system. And so we see increasing concentration of CAR-Ts here, and we see a decrease of that CD19 positive population, or CD19 positive CARs, CD19 CARs, and that CD20 population is stable. Eventually, what we want to do also is then add in small molecule screening to this so we can see, can we increase activity of the T cell population, of the CAR population, or control it and decrease it as well. And so this program is just getting off the ground, and we're hoping to add about 5 more samples into this in the coming month. All right, cool. So I want to change gears slightly and talk about 3D imaging. And I've seen a couple cool 3D images so far in this conference. And so what I said before, so we moved into 3D imaging for our solid tumor programs, and also one could imagine that we eventually move into it also for our hemato-oncology programs, especially in lymph nodes. And this is really made possible due to deep learning-driven image analysis. And so what we're looking at here is a Z stack through solid tumors or solid tumor cells, and this is overlaid with the segmentation and the classification in gray, yellow, and red. Here we have one of our microaggregates. So again, these pieces that came out of the solid tumor here in 3D, and then a reconstruction of this microaggregate. So we believe that these microaggregates are really quite interesting biologically, especially when looking at solid tumors or what's happening in the lymph nodes. We can start to begin to understand how these cell complexes occur or where they are, and then how drug changing these. Is there more activation and some of them less activation? Are we getting potentially, one could imagine, CAR T integration into some of these as well? And so, we haven't really gotten that far yet, but, we're starting to look at, okay, what else can we image rather than just 2D planes? And we have this nice spatial paper from 2017, but can we also then do this in 3D? So to show you just some examples by eye of the solid tumor screening program. So this is ovarian cancer. This is a 7-point dose response curve, 5 drugs I pulled out here. These are the responses, a relative response of the target cell population. And then here you can see the viability diet and the segmentation here. As we increase the concentration over, we see this drug is not really that active. This one also looks not that active. And these 3, you can start to see the volume of these cells change or more cell death in the images here. And these correlate with a drop in the relative response or the relative cell population of the target cancer population. And you can see the increase of viability die here in green. What's interesting also is you have this drug here that's not selective. It's a relative cell fraction normalized to the cancer burden. So this stays straight, but you start to see viability die here pop up. So this is an example from ovarian cancer. We're working also lung, breast, and head and neck next. But I think we're just beginning to scratch the surface of what we can measure from solid tumors utilizing 3D imaging and deep learning. And one of the things that really excites me is just the physical attributes of these cells, and we start to look at images of them. So this is some example images: DMSO, drug 1, drug 2, drug 3. And we can start to look at, here's the viable malignant cell here, here's an immune cell. So it's not just counting the cells, but it's also looking at how they're shaped and how they're formed. And here's an attackment, a potentially an engulfment. And so what we want to do is we want to determine if we can track how cells are dying based off their morphology. Pyroptosis, apoptosis, and necroptosis are very, let's say, very feature-rich cell death mechanisms. So we can start to look for these or add on orthogonal cell painting or metabolism tracking onto this, and then eventually begin to cluster these responses, not necessarily by expression of different markers, but by their visual phenotypes. And just as an example of this, here's a variation autoencoder type architecture clustering by visual phenotype, larger cells up here, smaller cells here. So unsupervised learning of clustering by visual phenotype and how these may change after drug treatment. And this will be quite interesting. So in the last few minutes that I have, I want to talk about something a little bit different that I wasn't necessarily going to touch on, but unknown to me, Baron mentioned a term yesterday in his talk, in his conclusion slide, patient-centric drug discovery. And I wanted to talk about our interpretation of what this could potentially mean in the translational medicine space. So this is what I want to show you is more patient-centric target discovery. But this is 17 samples from ovarian cancer patients. It's malignant pure effusion and ascites. It's not solid tumors. I'll give you an example in a second of that. Over 80 small molecule drugs, most of them approved for other indications or ovarian cancer. 7-point dose response curves and technical triplicate. And we have a cluster of small molecule drugs, actually a family within the same target space that are having strong on-target effects to the cancer cell population within a subset of these patient samples. Not all of them, it's not a cell line, but in a small subsection of them. This is malignant pleural effusion and ascites, so a secondary malignancy, cancer cells, immune cells. people ask, okay, well, is it possible also in the solid tumor? Yes, or so orthogonal confirmations of findings in freshly obtained surgical specimens of ovarian cancer patients. We see the same responses that we had found active in the malignant pleural effusion in ascites, at least in some low ends, but we're getting there. And so the idea here is that we start to see patterns of the data in, again, responders and non-responding patients. And we can, again, add orthogonal data on top of that in genetics and try to understand, look, these drugs are approved for other indications. Are we hitting the same target space? Do we find off-target activity of these drugs? What pathways are they affecting? And we can start to really understand the differences in the patient backgrounds and how the patient backgrounds genetically drive their response characteristics that we're seeing. So it's really, again, using drugs as tools for targets, let's say patient-centric drug discovery or patient-centric drug development and design. And this is something that is currently also still ongoing. So this already brings me to the summary and outlook. Again, off topic, but I wanted to be relatively short. So summary, big picture. So the work in the patient-centric translational research, we believe, is really driving our understanding, again, not just of the models and how to use them, but of in vitro diagnostics and precision medicine in general. The more we understand about our platform, the more we understand about our samples, the better we can push forward into the clinic. For microscopy for solid and liquid biopsies is occurring. Deep learning-driven image analysis enables really robust 2D and 3D single-cell quantification in very complex images. Fast, sample efficient, compatible with low tumor content and low cell content. And we know this from limited detection studies. Small molecules, biologics, and cell therapies. And we do quite a lot, or we try to do quite a lot of work on indication-specific technical performance evaluations to really understand, again, when we're working with material in our platform from many different study centers, how is it working and how can we compare this data together? And also it's gearing up for a US in vitro diagnostic drive in 2022, and we're looking for interested clinics. And we already started going out to KOLs and approaching different clinics. So it's, so the more that we can do between now and 2022 and understanding our platform is big. So I just want to say thank you to everyone at AllSite. We're 25 people and growing. There are some pictures from a hike we took in October. We grew quite a bit in 2020, and we don't have any proper group pictures because we haven't gotten together. But I want to say a huge thank you to everyone involved in the work. We have about 65 PIs around Europe that our clinical operations are working with for our sample collection. And a big thank you to key academic and clinical collaborators, Baron Snyder and Julius Frigert in particular, for whom we would not be here without. And then also collaborators I showed Medical University of Vienna, Uli Jaeger, Philip Staber, and Christoph Kornaut on the EXALT-1 trial, and Heinz Sill. I showed data from him too, from Medical Graz, along with our other collaborators from our Austrian study centers. So with that, thank you so much for the time. I very much appreciate the leeway in going off topic, and I'm happy to take any questions.